Welcome back to the Blood Cancers Blitz. Top 10 tools and keys to flow cytometry. We'll again begin with the question, how do you identify a hematologic malignancy? Pause the video and consider. I've noticed that it takes three things to identify a hematologic malignancy. First is an abnormal phenotype. Second is clonality. The third is doing bad things. If we have all three of these features, the WHO classification system has a box to put the entity inside of, and we can give it a diagnosis. But did you realize that the WHO classification is a work in progress? The current form is the best we have with the current tools that we have. Which begs the question, what are the tools of hematopathology? This was the challenge from yesterday. What did you find? How did you organize them? Here is my way of organizing the top 10 tools of hematopathology. I've summarized them by analysis target and analysis type. If we look at the targets, there are several targets we can consider. The whole hematopoietic system, cell morphology, protein markers, chromosomes or entire genes, parts of genes or base pairs, going from large to small. Different analysis types can be single or multiple. If we consider the whole hematopoietic system, a snapshot that we can acquire is the CBC, the complete blood count. From that, we can tell if there are increased lymphocytes, increased blasts, the number of other white blood cells like neutrophils, platelets, and red blood cells. All of these are important in coming up with a hematologic diagnosis. If we look at all of these features over time, we construct the clinical history, where we can look at trends within the white blood cell count to see if there is a persistent anemia, for example, or a persistent leukocytosis, or a persistent thrombocytosis. All of these are important for landing upon a diagnosis. The second row, cell morphology. We can look at this from a single cell perspective, for which we use the general term cytology. So the size of cells, the cytoplasm, the nucleus, whether there is any nucleoli, the texture of the chromatin. And if we look at multiple cells together, we will call that architecture or the arrangement of cells. Are the cells in question clustering together, forming diffuse sheets, or are they scattered? Moving down, we can look at the protein markers within the cell or on the cell. If we do it one at a time, we'll call that immunohistochemistry, where we can use different markers such as CD20, CD3, CD10 to look at one cell and one marker. If we look at multiple markers at the same time, technique that we can use is flow cytometry, where we look at multiple markers within the same cell. And we look at multiple markers with multiple cells. If we dig deeper into the nuclear material, we can identify chromosomes and entire genes with fluorescence in situ hybridization, or FISH. With this approach, we can identify one gene at a time and see whether it is intact or whether it's broken apart into smaller pieces and gets split up and sent to another part of the genome. If we look at multiple chromosomes, the technique is called a karyotype, where we'll display the entire genetic material by chromosome number 1 through 23, and then X and Y. Or 1 through 22, and then X and Y. Going even smaller, we can look at parts of genes or base pairs. We do it one at a time. We can use the tool of PCR, polymerase chain reaction, to analyze a target base pair within a target gene. And if we look at multiple genes or multiple base pairs within the DNA or RNA, we can use the tool of next generation sequencing, or NGS. All these together are the top 10 tools of hematopathology. Other areas in pathology, such as surgical pathology, may have other tools that they use, such as um, um, uh, methylation of DNA. However, within hematopathology, that is not one of our major tools that we use yet. If we look at these top 10 tools, we can actually compare them to the initial three characteristics that I mentioned before, doing bad things, abnormal phenotype, and clonality. And we can see how each row can correlate to one of the features. CBC and history, that helps us prove that a process is doing bad things within the patient. Cytology and architecture can also prove that it's doing bad things if it's taking over a place where it should not be taking over. Now, abnormal phenotype, we can see with immunohistochemistry or flow cytometry. And clonality, we can prove by showing different rearrangements of chromosomes or genes using FISH or karyotype, or even looking at parts of genes with PCR or next generation sequencing. With these tools are the transportation that we use to navigate throughout the Hemepath world and will help you go from one place to another in your diagnostic journey. With that, we're going to focus a little bit more on flow cytometry, as I have received um, feedback that flow cytometry is uh, one of the tools that seems to be complicated for people. Again, is one of our top 10 tools. Flow cytometry is an analysis that allows us to look at protein markers and multiple markers within the same cell. For this next part, our outline is going to be your digital eye, mindset shifts, and then give you a step-by-step process to help you walk through flow cytometry. Primary goal of flow cytometry is to be your digital eye, to save time at the microscope, to be an artificial or digital eye so that it can replace looking at multiple H&E sections or right gim sustains, followed by comparing um, multiple separate IHCs. So let's do this step by step. What do you see in this picture? You see red blood cells, platelets, and white blood cells. The white blood cells that we see, you probably quickly identified as neutrophils and a lymphocyte. However, how can we describe them in further detail? We see four white blood cells. Three, the neutrophils, are large with pink cytoplasm and lobulated nuclei. The lymphocyte is a smaller cell with scant cytoplasm and a round nucleus. Your eye recognizes this without even thinking about it. You just automatically notice that the cells look different. Some are larger than the other, and some have more convoluted nuclei than the others. What do we see here? Again, red blood cells and platelets, but the white blood cells are different. See two of the smaller cells with round nuclei and scant cytoplasm, and one of the larger cells with pink cytoplasm and convoluted nucleus. Again, your eye recognizes this automatically. What about with this picture? How many of each type of white blood cell are there? It's a little bit more challenging when there's more cells to consider. But this raises the question, is there a way to do this faster? As mentioned before, your eye automatically notices that there are larger white blood cells and smaller white blood cells. Your eye automatically notices that the cytoplasm looks different between the two cells. Your eye automatically notices that the nuclei are different between the two types of cells. And so let's keep track of this. We compare large cells versus small cells, and cytoplasm, whether there's granules or not, or the color of it, and the nucleus. We can combine, or we can transform these characteristics into size, that's large versus small, and then the texture of the cytoplasm and the shape of the nucleus, we can combine into a term called internal complexity. We made a graph of size versus internal complexity. We could easily plot these 4 white blood cells on this graph as shown. The neutrophils are larger in size and higher in internal complexity, and the lymphocyte is smaller in size and lower in internal complexity. And since we've already represented what they look like, we don't have to have the actual pictures. We can replace them with a dot. So is there a way to organize the white blood cells from this picture a lot faster? Yes, there is. And we can convert it to a chart that looks like this, where we see 12 neutrophils that are larger in size and have higher internal complexity, and 3 lymphocytes that are lower in size and lower in internal complexity. Now, these terms, size and internal complexity, we can convert to forward scatter and side scatter. These are the terms that are typically used in flow cytometry, but that is what they represent, size and internal complexity. Let's do this for another case. Here, the lymphocytes look pretty similar. They're all small in size and small and have little internal complexity. But this begs the question, what else can we do? If we were looking at this within a surgical pathology world, we could raise the question of where are the B cells and use immunohistochemistry to try to answer this question. Using a blue counterstain and a brown immunohistochemical stain, we could use an antibody against CD19 to identify B cells. And then in another slide, use an antibody against CD3 to identify T cells. Then ask where are the abnormal markers and use CD5, for example. And putting these together, we can make the conclusion that B cells have aberrant CD5. However, is there a faster way to do this? Using individual IHC, we had to compare between CD19, CD3, CD5, back to CD19, comparing slide and going back and forth to figure out which population had which stain. What if we did this within the same slide? Say we used one color for CD19, represented by a yellow triangle, another color for CD3, represented by the blue square, and a third color for CD5, represented by the red triangle, red diamond. We can then make plots or charts of one marker versus the other, say CD19 versus CD3 or CD19 versus CD5, and use some fluidics, lasers, and optics to plot all of these cells on a chart. Here's one way where we isolated CD19 positive cells or expressing cells compared to the CD3 expressing cells. And we can have another iteration where we look at CD19 versus CD5. With this, flow cytometry is our digital eye. It converts multiple separate IHCs into charts that represent the same thing but are easier to look at without having to flip slides back and forth. So the multiple slides on the left compared to a couple charts on the right. That's our basics of flow cytometry. Next step is to consider some mindset shifts for flow cytometry. The first one is that numbers are names. There are a number of CD markers that are used to identify different cells, such as CD20, CD3, CD5, lots of numbers. And it takes a bit of intentional practice to adjust, especially for English speakers, because we typically think of numbers as very separate from names. Some other languages might be a little bit different, such as in Hebrew, where letters can represent numbers. However, for English speakers, it takes a bit of time to adjust so that you are more fluent in the numbers that are names. The second shift for flow cytometry is understanding that flow cytometry is a tool, not an answer. It simply helps you see what you're looking for. And so to say, what did the flow say, makes about as much sense as what did the binoculars say. It really depends on where you're looking. Did you find what you were looking for? In a similar sense, saying that flow is positive is pretty imprecise because it doesn't specify what markers you're looking for. And so it'd be more precise to say flow cytometry demonstrates a population that expresses X, Y, and Z. We'll take a short break and admire nature for a second. But while we're here, have you thought about what it takes to weed a garden? Pause the video for a couple seconds and consider. First, you have to know what is planted. To weed a garden, you want to make sure that you don't pull up the things that are supposed to be there. Second, you have to acknowledge the limitations of your task. When you're weeding a garden, you're not paying attention to the air above or the roots below necessarily, or the components of the dirt. You're looking at the plants. Third, you probably want to check that the plants are normal. They're not wilting, they're not moldy, they're not being eaten by different animals and so forth, and that they're all intact. And then finally, oh, yes, so make sure the plants are intact here. And then finally, look for weeds, the things that are not supposed to be there. We can tell a difference in leaves here in the red solid circled area. Those are different than the green dotted circled area. The green dotted circle are the plants that we expect to be there, that we planted there, perhaps beets in this particular picture. And then the red solid line, or red solid circle, are plants that we did not want in our garden. Interpreting flow is actually like weeding a garden. First step is to be aware of what is supposed to be there. Second, acknowledge the limitations of the task. Flow cytometry usually ignores granulocytes and misses large cells. Third, check for the things that to make sure that the cells are normal. So looking for normal markers and then the presence of abnormal markers. And then look for weeds, whatever is not supposed to be there. You check for any unaccounted for populations. This can be expanded into a 7-step process where you first identify the specimen source, then identify the clinical question, list the expected cells for that source, list the expected markers for each cell and noting any overlap, check for normal markers, check for abnormal markers, then check for any unaccounted populations. Using this step-by-step approach, you can analyze flow from any source. Let's apply these steps to the case that we looked at earlier. Step 1, identify the specimen source. This is peripheral blood. 2, identify the clinical question. Question is this lymphocytosis neoplastic? Step 3, we'll list the expected cells for that source. What cells do we expect to see in the peripheral blood? And what cells do we see within the picture. In this picture, we see T cells, and we would expect to see B cells and T cells within the peripheral blood. Step 4, list the expected markers for each cell, noting any overlap. For B cells, we would expect to see CD45, CD20, CD19, among others. For the T cells, we'd expect to see CD45, CD3, and CD5. Notice that CD45 is expressed by both sets of B cells and T cells. Step 5, check for normal markers. In our charts, we had CD19 to pull out one population and CD3 to pull out the other population. CD19 is normally seen on B cells. CD3 is normally seen on T cells. So check and check. CD19 matches B cells and CD3 matches T cells. Then we check for abnormal markers. We use CD5 as an abnormal marker. In this chart, CD5 is expressed on the CD19 expressing cells, which is abnormal for B cells. However, CD5 is expressed in the CD19 negative cells, and that matches our T cell percentage. CD5 is a normal marker for CD3, for T cells. Finally, check for any unaccounted populations. As we review our initial charts, there are no populations that we have not checkmarked. And so we have accounted for all the populations. After you go through these 7 steps, then you can give your interpretation. We do have an abnormal population. B cells are aberrantly express CD5. We would suggest correlation with some type of clonality studies. The T cells are unremarkable. They retain normal expression of CD3 and CD5. So flow cytometry saved us time at the microscope, converting multiple IHCs to 2 charts, which we could then interpret to give a descriptive diagnosis, or to give a description of the populations. That has been a review of flow cytometry, but it then begs the question, which tools are the most important? The answer is that it depends where in the Hemepath world you are. And so we will now begin a tour of different parts of the Hemepath world and focus on how to apply the tools to navigate within each portion of the Hemepath world. Our challenge for tomorrow will start with acute myeloid leukemias. How many of them are you aware of? How could you organize them? Store your responses in a notebook, digital file, or blog. Bring back your answers tomorrow, and until then, let's blitz.